This is Science Chapter 2, Class 8, The Invisible Living World Beyond Our Naked Eye. Here in this chapter, as the name itself indicates, we are going to see the invisible world around us. I hope you all will enjoy this chapter. See, the human eye has a capacity to see certain things which have a size uh, above a certain size, and which are below the certain size means we are not able to see them. In order to see them, we need the help of microscope, and such particles are called microscopic particles or microscopic organisms if they are living. So we are going to first study about the historic inventions and discoveries, historic movements that led the people to discover this microscope, and all about the microscopic organisms. See, the human eye can see objects that are above a certain size. If they are too small, we are not able to see them. For a long time, many tiny things around us remained unknown because in those time people were not knowing about microscope. Nobody invented microscope at that time, and they were not knowing about that kind of a world which is beyond the capacity of their naked eye. And after so many years, people discovered or people started using a small piece of glass which was in the shape of a lentil seed, and they named it as lens. You know the shape of Dora cake, isn't it? It is thicker at the middle and thinner at the edges. That kind of a shape it was having, and people used to call it a lens. And they were able to see a few tiny things, and days after days, years after years, science progressed a lot, and now the tools which are using to see this kind of microscopic things are highly powerful. The invention of the microscope opened the fascinating hidden world filled with tiny living creatures. Now we are going to explore some of these life forms in this chapter. Some of the tiny microscopic life forms in this chapter. You have already learned about the amazing variety of living beings. Just look around. There are so many beautiful plants and animals. They are all different shapes, sizes, and colors. Some living beings are very tiny while others are really big. They differ not only in their structure but also in many other features. All these living beings, whether plants or animals, are called organisms. When we look around ourselves, we will be able to see n number of plants and animals. They are having different shape, different colors, different features, different adaptations. So we call them together as organisms. Have you ever noticed the smallest organism around you that is visible to the naked eye? When we can look through our naked eye without the help of any kind of aid, can you tell me which is the smallest creature around you that you can see without any kind of devices, using your naked eye only? Think about it. How small a thing can your eyes actually see? Your eyes may be highly powerful than my eyes. I can see an ant, but you may be able to see the organisms which are smaller than ant itself, isn't it? So you might have seen some people using reading glasses. Your father, your mother, or grandparents, they may be using reading glasses. Even you yourself may be using reading glasses due to any kind of defects of your eyes. How does it help them see better? How does this glasses, reading glasses, help the people to read better? Have you ever thought of that? Or what happens when we use a magnifying glass to observe something? In your chemistry laboratory or biology laboratory or physics laboratory, wherever you go, you will be able to find a magnifying glass. So when the next time when you go to the lab, you please take a magnifying glass with the permission of your teacher and observe how you are able to see the things by this magnifying glass, or when you look through the magnifying glass, how you are able to see the world around you. Now next we are going to explain an activity. Let's observe. Take a round bottom flask made up of glass and fill it with water. Here in this picture you can see a round bottom flask. The bottom side of the flask is round, that's why it's called round bottom flask, and it is filled with water. And behind this flask you can see one textbook is kept. It is your class 8 old textbook that is kept there. And when you look through this glass you can see that the letters seems to be bigger in size. So this round bottom flask, the flask which is filled with water, is acting as a magnifying glass. And if you are looking through this glass an ant, you will be able to see the ant very clearly. The different parts of the ant can be seen very clearly. So for long time people were curious to explore. This curiosity was there in that time also. They were highly curious to know the world around them, but they could not see them because in such days no sophisticated microscope or such kind of things were not invented. So how did we finally discover this invisible world? Which discovery or invention led the people to see this invisible world around us? We are going to study the story behind this discovery of invisible world. Ever heard of? You can explain, we can just explain this historical part. In 1665, a scientist named Robert Hooke, underline the scientist name, it's very important, Robert Hooke, and the book written by him is called Micrographia. Actually, this book consists of several drawings made by Robert Hooke about the small tiny things that he observed through his magnifying glass. He was a careful observer and at the same time he was a great skilled artist. So he drew several kinds of drawings which are related to the tiny organisms that he saw using his magnifying glass. His microscope made things look 200 to 300 times bigger than their size. So while he was looking through his microscope, he could observe the things which are bigger in size, bigger than their actual size. How many times? 200 or 300 times bigger than their actual size. One day he looked at a thin slice of cork and saw it was made of many small empty spaces. One day he was observing a dead wooden piece, or what we call the cork, under this microscope, and he could see so many rooms like structure, compartments like structure, empty spaces were there, and he called such compartmentalized empty spaces cells. This was for the first time somebody used the word cell in the history of science. Around the same time in 1660s, Antoine van Leeuwenhoek, a Dutch scientist, made better lenses that allowed him to build more useful microscope. The scientist's name is Antoine van Leeuwenhoek. He was the first person to clearly see and describe tiny things like bacteria and blood cells. He was the first person who could see, who could observe a living cell for the first time. What Robert Hooke observed was non-living cell. It was a cork cell, a dead one. But the first person who observed a living cell is Anton van Leeuwenhoek. He observed bacteria and blood cells. Because of this, he is known as father of microbiology. Micro means tiny, small one. That is the biology which deals with microscopic organisms. Small, tiny organisms. That branch of science is called microbiology. Microbiology means biology of microscopic organisms, microorganisms. That branch is called microbiology. So who is known as father of microbiology? A Dutch scientist called Anton van Leeuwenhoek. Here, Micrographia book, you can see the microscope of Robert Hooke. This is the microscope of Robert Hooke, and this is the compartments that he called the cells he observed through his microscope. Next one, what is a cell? What is a cell? This question is very important question, and so many times, several times you might have heard this word cell. All living things are made up of cells. You might wonder what cells actually look like. Let's take a closer look at the basic structure of a cell using a microscope. So, cells are the basic units of all living things, whether they are plants, animals, their body is made up of cells. Our body, plants' body, animals' body, microorganisms' body, all the living structures are made up of cells. So, cells are called the basic functional and structural unit. The basic structural and functional units of living things are called cells. Again, one activity is the that will help us to observe the cells under a microscope. For that purpose, we need to take an onion peel. Onion you can take, onion is easily available at your kitchen, so you can take this onion to your lab and watch or observe under the microscope how a cell look like. Take an onion bulb from your kitchen or garden and wash it thoroughly with water. After washing, we will cut the onion bulb vertically into pieces. We will make into several pieces. Take one piece of onion and pull out the thin transparent layer from its inner surface with the help of a forceps. Like this, you are able to easily just take a peel. A very tiny layer is there, very thin layer is there. When you cut the onion, you will be able to observe this thin layer, that using a forceps we can just take this layer out. This layer is called the onion peel. Place this peel in a petri dish. This is called the petri dish. You can see a round colored dish is the, this is called what? Petri dish. We can just keep this peel in a petri dish containing a few drops of saffronin. Saffronin is a dye. It's a stain. Red colored stain it is. We want to stain the cell, otherwise we'll not be able to see it. That's why we are giving color. In order to give it color, we are keeping it in a colorful substance, red colored substance called saffronin, for 30 seconds. This will give a pinkish color to the cells and help us see them clearly. Now, with the help of a thin brush, you can see with the help of a thin brush, this person, this picture you can see, he is just picking it up and it is placed on another petri dish and containing water to rinse the peel and remove extra stain. See, first we will put this onion peel in a petri dish which contains saffronin. What is the need of saffronin? To give it a red color or pink color, so that it will be easily visible through microscope. Now excess color should be removed off. It should be washed off. It should be thrown away so that everything will be clear. So for that purpose, we will just keep them in another petri dish which contains water and just wash it to remove the extra color. Now carefully place the stained onion peel on the glass slide using a thin brush. This is the thin brush, and using this thin brush, the person is just keeping this onion peel on a glass slide. Ensure it does not break or fold. You should be highly careful while doing it. Put a drop of glycerin over the onion peel on the slide. Now we can just put a drop of glycerin over the onion peel. Why glycerin? You know, glycerin is a highly moisturizing substance. So it should not dry. The onion peel should not dry. To keep it moisturized, we are adding a drop of glycerin. Why we are adding glycerin? To keep the piece moisturized. The glycerin will prevent drying of the cells and improve clarity for better visualization of cells. It will never allow the onion peel to dry up, and we will be able to see the cells very clearly. Slowly place a cover slip over the peel using a needle such that no air bubbles get trapped. Carefully, we should just place a cover slip. You can see how the cover slip to be placed. The cover slip which is kept 45° at an angle, and it slowly we will just remove this dropper and place the cover slip without making any air bubble inside that. There should not be any air bubble inside the cover slip. Now it is placed very nicely. Now view this slide under a microscope or a foldscope. Now it is ready for us to observe. Now we can see it under a microscope. You will observe nearly rectangular structures under this microscope. This is the way it gives you. These are the cells of the onion peel, which are closely arranged without any space in between them. They are very closely arranged. You can see the square shaped structures, just like bricks they are. How the bricks are arranged in a building, the cells are arranged to form this onion peel. Try to observe the peels of the leaves of different plants around you. You will find that all plants are made up of cells. What do you think the body of an animal is made up of? Of course, the body of an animal is also made up of this kind of cells. See, a comparison is given here in between the bricks and this what we call the onion cells. Just like the bricks arranged, the onion cells are also arranged. Here we can see the different parts of the cell. The liquid part inside the cell is called the cytoplasm. The highly colored structure, the highly dense part is called the nucleus, and there is a boundary called the cell membrane, and cell wall is there. And cell wall is seen only in the cells of plants, not in the animals. We cannot see cell wall in the animals. So this is the basic structure of a cell. You should mark the cytoplasm, which is the liquid part inside a cell. Then the most important part is the nucleus, which is called the brain of the cell, the control unit of the cell, which is darkly mentioned there. That is dark color, it is dark pink nucleus, and cell membrane is the boundary, and beyond the cell membrane you can see the cell wall. Cell wall is the extra membrane, that is after the cell membrane you can see this cell wall, which can only be seen in plant cells, not in animal cells. Now the next activity in your book is let's investigate. That activity is about how you are able to observe, or how you can observe an animal cell, how it is different from a plant cell. All these things we will be discussing in the next video. Next video will be beginning with the animal cell observation. How we are able to, in what activity we are able to see the animal cell under the microscope. So thank you so much. We'll be seeing in the next video.